If I go too fast, just do something. I tend to go.
Hear me all right? Great. Thank you, everyone, for coming this morning. And for those of you who don't know me, I'm Terry O'Sullivan, and I am a faculty member here at the University of Guelph in the Department of Population Medicine here at that college. And you guys are behaving just like the vet students and all sitting in the back of the room and not in the front two rows. So you're fitting in just fine. I'm delighted to welcome you to the first University of Guelph Swine Research Day. And as many of you know, this event has come about by the collaboration and the joining of the Centralia Swine Research Update, as well as the Mike Wilson Swine Research Day. And so the committees of those two events have joined together and formed a new committee to create this University of Guelph Swine Research Day. And the interest in today's event has exceeded our expectations, and so because of that, we did move the venue, obviously, here to the Vet College, because we outgrew the Arboretum. And I'd just like to thank the Dean's Office at OEC for accommodating that transition so on such short notice. I'm also very excited to tell you about our keynote speakers, Dr. Jim Squires and Dr. David Frazier, and they will be live podcast by our IT department here, so they'll be available online afterwards as well. And this new format has a new graduate student poster and oral competition. So the students they've been working hard on their abstracts and their oral and poster presentations, and so they will be judged, and we'll have some prizes at the end. And the posters actually are going to be in the room on the side here. We did have both rooms booked, but the room got double booked, and there's a defense in the other room, so I didn't want to stress out the candidate too much. So we just stuffed all the posters into the one room. So sorry about that being a little bit squishy there. And as with most conferences, this didn't happen magically, and there are many people to thank for their help and support of this initiative. And first of all, I'd like to thank our sponsors. We've had some very generous support from all of our sponsors, my team in total, which is just terrific. So if we could just thank the sponsors. I'd also like to acknowledge the organizing committee for this event, which is called Friendship, Kate Delaney, Judy Smith, Doug Richards, Jean Calvin, Bahal Farzan, Karen Richardson, and Carrie Parsons. We could thank everyone. And just one kind of key housekeeping items. If you did not park in P30 or pay at a meter, you're probably going to take it and maybe be towed. So if you're not sure where you parked, just ask someone that looks like a student or from the University of Guelph, because I arranged for you to be not ticketed, parked in P30. So just wanted to touch base about that as well. Yes, and so I have learned something new this week already. I have just recently opened a Twitter account, and so we will be following the conversation today on Twitter. It's in the program, and I'm still learning how to tweet, so I'm going to try to do that today. And now it's my pleasure to introduce the Dean of the Ontario Veterinary College, Dr. Jeff Mitchell, and he would like to say a few words.
Thank you, Carrie, and thanks to the entire organizing committee, which there were many members there, for putting on such a great day. What an amazing attendance. It's always a good sign when you have to move your venue to accommodate a larger crowd. So tremendous effort there. I'm fairly new dean. I've been here just 6 months, so I'm learning a lot about not just University of Guelph, but the livestock industries of Ontario. I have met with Ontario, it's very important. I'm very impressed with the industry here. I think the longevity of these research updates is something. It goes back to 1982, I believe, for the Centralia, I was told, pre-internet days. It's interesting to see, these days. I guess when the internet and the various other technologies came on board, I think, obviously falsely assumed that many of these face-to-face knowledge transfer events such as this one would become less popular and less relevant. Well, they certainly have. And over the years, and just in May this year, we've got a number of different events like this with different producer organizations. And it's just a tremendous sort of testament to the breadth of research expertise we have here at the University of Guelph and their willingness to give back to the industry and for the industry to support that. This type of innovation that we have in Guelph. So thank you all for coming. I think, judging by the program we have here, we have a number of fantastic researchers from over West presenting their results today, and I think you'll go away today with feeling a little bit inspired by the kind of research. Much of it is very applied, looks very relevant, and can be applied on swine farms almost immediately. So really impressed with that. I think Dr. Campbell is going to join us later on.
Yes, yeah. Dr. Campbell is this afternoon.
So Dr. Campbell is our VP for research, and as a member of the senior executive of the University of Guelph, I think he will give you a message, which I will give also, is that the University of Guelph is very much about returning to its roots in terms of supporting the livestock industries, agriculture, and food. I think we're sort of entering a new era, and I think it reflects also the importance of food and agriculture and health to society in general. So, the University of Guelph is definitely responding to that. The swine industry is extremely important. It's important to the province. I understand that it's responsible for over $13 billion in economic impact to the province. So that's not to be ignored. And I think you will find the University of Guelph and the different colleges will be very interested in continuing this partnership with the swine industry going forward. It's important for us. It's important for us to have our trainees trained in and around the problems that are important to your industry. And of course, it's important for your industry to have a group of researchers like this to help answer those difficult questions. So I think you're going to have a great day. Enjoy.
Thank you. And it's now my pleasure to introduce our first University of Guelph Swine Research Day faculty teacher presentation. Dr. Jim Squires is going to come up and talk to us about solving the boar taint problem.
I'm going to get your mic. Yep. Just yeah. Thank you, Terry. And I want to start off by thanking the organizing committee. It's quite an honor to start off a whole new series, research series, by being the feature speaker. When Bob Friendship asked me to talk, he didn't say feature speaker, by the way. And what he did ask me to do was actually to kind of walk through my research program on boar taint, and that's what I'm going to do. And actually, that's been a really fun thing for me. If you've been working at something for as long as I have, like 29 years, and you go back to the beginning, like what's the very first thing you did, and how does it lead up to where you are now? Wow, it's quite a walk. So I hope you enjoy it. So I call it solving the boar taint problem. We haven't quite solved it yet, but we're getting pretty close. So my background is I got my PhD in biochemistry in 1984 in enzymology. My master's was in acid-base balance. So I have a solid background in metabolism and enzymology. And after I finished my PhD, I went to some faculty advisors and said, so what do I do now? Because I have a PhD. Well, they said, you have the postdoc. Okay, so I applied for an NSERC postdoc, and I got it. So I went to actually National Institutes of Environmental Health Sciences. So one of the institutes of NIH in the US, I was there from 85 to 87. It was like going to Mecca. It was just really quite an experience for me. So then in 1986 or so, I started applying for jobs, and I landed assistant professorship here in 1987. I just want to show you. So this was the advert, see 1986 here, and I think Jeff mentioned about pre-internet days. This was pre-internet. So this advert was in the back of Science. That's where I saw the job advert. And so they were looking for a molecular geneticist in animal and poultry science. I came up in biochemistry. I never heard of animal and poultry science before. So I phoned Gordon MacLeod, the chair of the animal science department, and talked to him about it. And he said, oh, we'll teach you the agriculture when you come here. And I thought, what a great idea. Look, you can actually use biochemistry for something really applied. So I like doing basic science, but I like to see an applied end. That's why I'm here. So anyway, it's been a great ride. Been a lot of fun. Let me just kind of walk you through some of it. Anyway, here's me. I did have hair at one point. That's 1987. So I said, and I still have the card in my pocket, actually. They have never asked me for a new card. Anyway, I made professor in 1997, and as I say, my research interests are in metabolism and genomics. So my postdoc work, I was actually involved, I went to a lab in NIH that was actually inventing cloning systems. So one of the reasons I went there was when I applied for my NSERC postdoc, the Dr. Nagishi, Masa Nagishi, who I ended up going with, he wrote a little blurb about what I was going to be doing, and I didn't even understand it. So I said, I got to go there. This is going to be really cool. And it was. So I ended up in a lab that was really inventing cloning systems back in 1985. So that was very, very early cloning. And I actually cloned 2 forms of cytochrome P450 in mouse, got this nice paper in biochemistry and this nice paper in JBC, which I never would have been able to do without having that kind of exposure. I'll talk about this a little bit later about how technology drives research. But this particular work here, this is like 2 years worth of work. I could redo this paper here in a couple of weeks now using the technology we have. All of this cloning at this time was before PCR. I know anybody that does any cloning says before PCR. There was a before PCR, there was. And that's how we did it. We did it the hard way. So it took a while to get things done. Anyway, just so you know, cytochrome P450, I got introduced to it when I went there. It's a very interesting system. It's a family of mixed function oxidase enzymes, so it's a whole series of enzymes, like saying proteases or anything else. They're involved in the metabolism of a variety of endogenous compounds like steroids and fat-soluble vitamins. That's what interests me. It's also involved in the metabolism of xenobiotics, like activation of carcinogens, like PCBs, for example, and also in the clearance of drugs, like CYP3A4 is a major P450 isoform involved in drug clearance. And so here's what it is here. It actually ends up hydroxylating products, and here you can see it has a heme center here. So I landed the job. It was a molecular geneticist and they said, oh, we'd like you to work in poultry and swine. And that was all the guidance I had. So I walked around and talked to different people and said, okay, I'm working in sort of molecular toxicology. We're in animal production, food quality and safety. What are some issues that would make sense? And I ended up talking to different people, and I call it pharma meets farming here, sort of P450 metabolism to animal production, and realized that this is really quite a significant issue. So as you all know, pigs are castrated to prevent boar taint, but this castration decreases muscle growth and feed efficiency, and also raises animal welfare concerns. So the goal, of course, is to eliminate taint and prevent castration, which also improves efficiency of pork production. And there aren't many systems that will give you both improved animal welfare and improved productivity at the same time. So I thought, this is great. So I call it win-win. And when it comes to funding, we'll talk about that a little bit later, sometimes you get funding because of animal welfare concerns, and sometimes you get funding based on productivity. It's fine, either way. So just a little bit more. So boar taint, as you know, is an off odor and off flavor in pork from uncastrated, intact, or entire males, depending on where you are, what they call it. It's really caused by 2 different compounds. When I first started, it was really androstenone, a steroid produced by the testis at maturity. It actually acts as a sex pheromone, right? So it regulates female reproduction. When I first started, this was thought to be the true boar taint, but it turns out that skatol, or 3-methylindole, which is produced by bacteria degrading tryptophan in the hindgut, is also important because it's produced in both males and females, but it's cleared more slowly in males. So the liver metabolism is slower in males. So you have both of these compounds. And it turns out the metabolism of boar taint compounds involves cytochrome P450. When I first started, in fact, there was some question about that. And so the very first paper that I did was this one here, published in 1989 in Journal of Steroid Biochemistry, talking about the involvement of cytochrome P450 in that very first step in boar taint synthesis. So I was able to say, okay, this actually does involve P450. So now I've got a real application of my postdoctoral work to something that really has a practical application. So I kind of ran with it from there. Okay, so I have something to work on. Now, how do we actually build a program? And this is all in hindsight, of course. When I started, you kind of wing it. But just for those of you who are here and thinking about it, these, I think, are key aspects. You want to find good people, students, technicians, and postdocs, and I'll show you a list of some of those as we kind of go through. Absolutely key. Collaborators with complementary experience. So you collaborate with people because they can add something to your research program that you don't have. like I wouldn't collaborate with another biochemist, what's the point, right? But you collaborate with other people who can give you that extra, that other aspect. You use the latest technology, or as I'll show you, you invent your own and as necessary. And you keep up with technology too, as I'll mention later. This is key. You need sufficient continuous funding. So when I came here, I applied for an insect discovery grant. I've held one since I've been here in 1987. Pretty proud of that. Insect discovery is great. It funds basic fundamental research, but it doesn't give you enough to run a program. I would never be able to do what I've done on just insect discovery grant. It's industry and government funding that's really, really driven the research in the end. And this is where you need something that has a real practical application. So you need to have a blend of basic work and with a real application to it. And of course, you need to publish your results in both peer-reviewed articles and also in industry publications. So I have about 70 papers just on the boar taint area alone. I also, by the way, when I first came, just to back up a little bit, I not only started work on boar taint, but I started some work in poultry too. And just, I won't talk about that today, of course, but I think it's always good to start a couple of different things for those of you who are thinking about that, so that if something doesn't pan out, then you've got something else to fall back on. So I think it's pretty key. Anyway, okay. So if I look at the program itself, the overall goal is to control boar taint without castration. We're going to identify and measure boar taint compounds. That was the first task. We didn't have any tools for this when I first started. Really, the whole idea is to understand androstenone and skatol metabolism because, and identify factors affecting the accumulation of skatol and androstenone in fat, because the underlying goal is to develop methods to decrease boar taint. You want to solve the problem, right? So I'm going to start off by talking about identifying and measuring boar taint compounds. So when I first came, as I mentioned, there weren't any real assays for the boar taint steroids. So we had to develop our own, and this is the first paper that I published here in Canadian Journal of Animal Science, 1990, where I essentially developed a colorimetric test for boar taint. I'll show you a picture in a second. So this was the first paper, and then the second paper on that area was collaborating with Elizabeth Collette, Ken Fisher, and Gary Partlow here at OVC, on really showing that the colorimetric test that we have actually correlates with sensory analysis, trained sensory panels, because you had to be able to show that. So that was our first test. We now have an ELISA, and I'll show you that we've done some other development as well. So here's what the test looked like in the beginning. What I did to develop the test was just dig back in the old literature. Again, you know, you can recycle technology. And I found this paper in 1961 where they were measuring these boar taint steroids in human urine. So boar taint is actually made by humans as well, right? So I took the chemistry from this and adapted it so I could run the assay in both fat and salivary gland. And here's some data from that first paper that I mentioned, where I got, these are standard curves using both fat and salivary gland to show that for the amount of boar taint steroids added, I get an increase in absorbance at 590, which is what you're seeing here. So this was great to start with, but clearly we had to go from there, and I'll show you as I go along how we actually advanced that. So we had this colorimetric test. I actually had some other opportunities. I had a really interesting student, Wei Yuan Zhu, who was interested in electronics, and I just struck up a collaboration with Gord Hayward in the engineering school. And he was interested, he was working on these piezoelectric quartz crystals. And what we really developed was really a prototype electronic nose. And so some of you may have heard of electronic noses, really a sensor that will measure different compounds. And so the whole idea is that you coat this electrode here. This is a gold electrode, a quartz crystal here. And this crystal will resonate at a certain frequency. We coated the electrode with a C18 material, a very nonpolar material, so it would bind androstenone. And then the frequency of oscillations varies with binding. So we were able to, using this kind of equipment here, where I had a sample here, blowing nitrogen through up, we had a crystal chamber in here with an oscillator, frequency counter, and computer, able to show that the change in frequency with the increasing amounts of androstenone. So we actually had developed an electronic nose for boar taint. Unfortunately, due to funding, we weren't able to take it past this part. But I think this had some real potential. The other problem, of course, is that because you're looking at a vapor issue here, using this in a packing plant would probably be a problem. But I think the whole idea about using something electronic like that would be very cool. And before he left, the student Wei Wan, he kind of drew up this sort of futuristic idea of what a rapid test for abortion taint might look like. So you'd have this sort of gun where you put a fat sample in here, nitrogen blowing through, heat the sample, add the chemicals that are necessary. It would then stick onto the crystals here, and the frequency, the signal frequency to the signal output to a frequency counter would give you the amount. So we never did build this, but I think it's pretty cool. Okay, let me back up. So where are we now with this? So we have ELISA, as I mentioned. About a couple of years ago, I'm in the collaborative tox program here at Guelph, and there was a seminar that was advertised by Maria De Rosa from Carleton University about DNA aptamers. And at that point, I had never heard of DNA aptamers, but it turns out that they're very useful. They are actually synthetic nucleic acids with the capacity to bind target molecules. So essentially, a piece of DNA or RNA will make a three-dimensional shape like this here and be able to bind the target molecule. So they act like antibodies in a way, but they have advantages over antibodies. Of course, you can make them very cheaply. And once you know the sequence, then you have it. You don't need to make antibodies over and over again. They also have a long shelf life and can be reversibly denatured without loss of specificity. So we plan to use these aptamers to develop a low-cost biosensor for boar taint. And there are lots of different formats you can use, rapid ELISAs and so on that we're working on. So this is actually an ongoing collaboration with Maria De Rosa through a project that we have with Canadian Centre for Swine Improvement. So this is actually pretty exciting stuff. Okay, so move on then. That's where we are with measuring boar taint compounds, understanding scatol and androstenone metabolism. Here's a key step here. So androstenone, as I mentioned, is a steroid made in the testis. It's made from pregnenolone via this enzyme, the cytochrome P450, of course, CYP17A1, and it needs cytochrome B5 as sort of a cofactor. This is a branch point where pregnenolone is either converted to androgens, which are the normal sex steroids, or to androstenone, which is the boar taint. So for a lot of our work, we've actually been looking at how can we affect this switch? So we can have pregnenolone converted to androgens, but not to the boar taint steroids. That maintains a normal boar without any boar taint. So that's what we've been working on. And I've had a number of students that are involved. So here's the first paper. John Meadis, a first PhD student of mine, actually purified the enzymes from adrenal gland, from both pig and cattle, and put together the different components and actually showed what components were necessary to make the boar taint. So this was a very early paper back in 1993. A second paper by Sean Davis, another student. Actually, we did Western blotting here to show that the amount of B5 varies within the animal, and the amount of B5 actually correlates with the synthesis of those boar taint steroids. And then a more recent paper here in 2009 by MacTell-Billen. By this point, we had gotten into actually protein expression. So at this point, we were able to clone cytochrome B5, two different forms, B5A and B5B, and CYP17, and actually express them in the cell line. So we've moved from purifying the enzymes from tissue to actually expressing them recombinantly. And we do this kind of thing now routinely. So all of these papers really show that the cytochrome B5 is critical for actually making the boar taint steroids, and if we can modify this reaction here, we can prevent boar taint from androstenone. We also looked at the metabolism of androstenone. So androstenone is made in the testis, but it's metabolized in the liver. Actually, a lot of it that's made in the testis is actually as sulfa conjugates. So that means it's a steroid, but it has a sulfate group stuck onto it. So if you take a very nonpolar steroid and put a sulfate group on it, it makes it very, very polar. So now all of a sudden it doesn't accumulate in fat, right? So what Phil Sinclair showed for his PhD was that he showed that these steroids are released from the testis as sulfa conjugates. Actually, here looking at it in vivo, but here looking at it in cultured porcine hepatocytes and also in Leydig cells as well. And that the amount of sulfa conjugation affects the accumulation in fat. So now we have another control point that we can look at. If we can increase the amount of sulfation of steroids, we can decrease the accumulation in fat. So the whole idea of understanding this metabolism is to think, okay, how can I use this information to control the problem. And again, just putting up these 3 papers here again shows you how technology has affected it. So in the beginning here, we're working in a live animal, and then we moved on to actually primary cell lines. Okay, in 1996, I took my first sabbatical, went to Sweden, to the Swedish University of Agricultural Sciences in Uppsala. An amazing experience. Just want to, I call this the importance of sabbaticals, because what sabbaticals are an opportunity to learn something new, right, and retool. So I've been at Guelph for a while, went to Uppsala, and essentially I'll show you some papers we got from that. From that, we actually established that this particular form of cytochrome P450 was involved in skatal metabolism, and we actually developed an ELISA for androstenone using antibodies that was available there. So here are 3 papers that came from that particular one. You'll see it looking at P452E1 here. This was my very good friend and colleague, Kirsten Lundström, and a student, Jacob Babel, who was here, did his master's with me, and then moved on to SLU to do his PhD with Kirsten. So in these particular ones here, we were looking at the role of 2E1 here, and looking at metabolites that we could see in tissue samples and in blood. We moved on then with Mike Turner, master's in 2005, again looking at cultured porcine primary cells, hepatocytes, and looking at the using different inhibitors of P450 to show which ones were involved in skatal metabolism. And then, as I mentioned, by the time we got a little bit later, 2011, Paulina Warchinska used the same technology as I mentioned before, where we actually express the different cytochrome P450 enzymes recombinantly in a cell line, either with or without cytochrome B5A, and showed from that we could actually definitively show which enzymes were involved in skatal metabolism. We also looked at what are the pathways for skatal metabolism, and this was work by Gonzalo Diaz, who was a PhD student of mine in 2000. And essentially what he looked at were what called phase 1 metabolites, which are all these ones that are shown here, and also the phase 2, which is the conjugation reactions that occur afterwards. So he did some pretty seminal work here and published it in Drug Metabolism and Disposition and Xenobiotica. You'll see that I have a real mix in journals that we use. More fundamental work like this goes in these kinds of journals, and the applied work goes in the animal science journals, right? So, and you do that to really target the audience that you're trying to reach. Okay, next part is about identifying factors affecting the accumulation of skatal and androstenone in fat. And this was work done by Franz Lancier, a master's student, and again, some work by Mike Turner, looking at how development affects skatal metabolism. So looking at skatal metabolism in very young pigs and very old pigs, and then looking at how that relates to enzyme activity in the liver and concentrations of skatal and plasmin fat. So we learned a lot of fundamental ideas from this work. And again, you see this work is essentially livestock science and Journal of Animal Science. Phil, again, for this is going back when he did his master's, actually. So he did his master's and stayed on to do his PhD. He looked at how androstenone changes during development here, and there's some interesting work here where he looked at the levels of androstenone that you'd see in plasma of pigs over time. So in the very young pigs that are about 2 to 3 weeks old, you get a peak in steroidogenesis here, which then flattens out and then goes up again at puberty. What he did in this work was he used a gonadotropin-releasing hormone agonist, leuprolide, that he injected the pigs with here, and he was able to blunt this first peak in steroidogenesis here. We were hoping at the time that we would see an effect here later on, but it didn't happen. And you see Dr. Rayside, Jim Rayside's been quoted in several of these papers as well. He was an extremely great mentor for me when I came to Guelph. Here's another paper by Phil where he looked at the amount of androstenone in fat compared to bulbourethral gland length, so really sexual maturity. And what he showed was that you had to have a certain length of bulbourethral gland, about 11 centimeters, before you had any animals here that had high levels of fat. So essentially, immature animals don't have vortein from androstenone. So he was using this as a pre-screening tool, and this is 2 papers in 2001. Okay. Now I want to talk about my second sabbatical, which is in 2003. I went back to the same lab that I did my original postdoc in, same PI. At this point, I'd been doing things At this point, I hadn't even run a PCR, believe it or not, in 2003. So I needed a complete retooling in molecular biology techniques, so I went down there by myself. My youngest daughter was still in high school, so I went to NIH by myself and spent 10 hours a day in the lab. It was a blast. And I learned all the latest molecular biology techniques, cloning, protein expression, reporter assays. I already mentioned about the protein expression assays. That's where I learned them. And the latest cloning techniques with PCR this time. And at the time, Nagishi's lab was looking at the role of nuclear receptors in regulating gene expression. And so I got into that. And here's a paper that I published, another paper in JBC, where I was looking at this pregnane X receptor, PXR, and looking at parts of the receptor that were involved in localizing it in the cytoplasm versus the nucleus. Again, I can't really emphasize the importance of sabbaticals. When you're in one place working for a while, some of the technology just passes you by. And you need to go to a place like this. And Tatsu Sayoshi, good friend now, was really my mentor, or my tutor, I should say, when I went there. He sort of held my hand for a while, walked me through a lot of these techniques, and at the end we ended up publishing a nice paper in JBC together. Pretty proud of that experience. So I came back here. Actually, while I was there, I was supervising my students sort of long distance. John Peacock, a master's student, had worked on so for my sabbatical work, the pregnant X receptor I was working on was in mouse. So it's NIH, so you're looking at inbred mouse lines, that kind of thing. Turned out that these receptors hadn't been characterized in pig at all. So I thought, great, this is a great opportunity. So the first paper here was characterizing the porcine receptor CAR, constitutive androstenedione receptor. And then the second paper here looks at pregnant X receptor, PXR, and also FXR. And this was work by Matthew Gray, another student who did his master's and then subsequently PhD. This paper here was published in Experimental Biology and Medicine, and I'll show you in a minute why that was quite important. After we characterized the receptors and their ligands and whatever, Matthew then, for his PhD, looked at regulation of actually steroid hormone synthesis and gene expression in both Leydig cells and hepatocytes, right? So he was able to really characterize the importance of these receptors in regulating boar taint metabolism. So here's where we are with, sorry, let me make sure I didn't miss anything, okay. So here's where we are. We were able to develop from this. We have 3 nuclear receptors that we looked at, CAR, PXR, and FXR. We have androstenedione metabolism in this box, skatal metabolism here, and all the arrows show that where we actually demonstrated that the enzymes in these pathways were regulated by these particular receptors. Again, why is this important? It's important because if you know all these pathways, then you have a whole bunch of targets you can shoot at if you're trying to regulate metabolism. So, for example, we could use different ligands for receptors to either turn them on or turn them off, right? I mentioned about the paper in journal in Experimental Biology and Medicine. After we published that paper, this chap here, Doug Buren from Baylor Medical College, phoned me up. Doug Buren uses preterm, he's interested in preterm human infants, and he uses preterm baby pigs as a model for preterm infants. And these infants and pigs are actually fed intravenously. And it turned out that the solutions that they were using were called, if they're on these total parental associated total parental nutrition for a period of time, they develop fatty liver and other complications. So Doug wanted to try to refine these lipid emulsions that were used in this condition here to try to eliminate that problem. And the reason he called me is because we cloned PXR and FXR, and he wanted to know how those particular compounds, in particular phytosterols and vitamin E, affect gene expression through FXR and PXR. So he was doing the physiology work, and we were doing the fundamental gene expression work here. And so we have some, again, papers in Journal of Lipid Research and parental and enteral nutrition journals. And this is just in the last couple of years. So it really shows that, well, the reason I show this is because, you know, in animal biosciences now, we do work on animal productivity and animal products, but we also use commercial animals as models for humans. And this is just a great example of that. And in our department, we have several other examples of similar kinds of work, but I just wanted to show you this. Okay. So we're finally getting down to develop methods to decrease boar taint, and the three key ones would be immunocastration, some dietary strategies, and genomic selection. And I want to show you what we've dabbled in those. The first one is immunocastration. You probably all are aware that there's a commercial vaccine available now, Improvest from Zoetis. It's actually an anti-GnRH vaccine, so you block GnRH here with antibodies, and essentially you are then blocking the function of the Leydig cells in the testis, so you're blocking the synthesis of both androstenone and the sex steroids. So it's an available vaccine, but it's currently not well used in North America and Europe. I think its use is increasing, and it's certainly a very good product. It works very well. This paper that I put up here wasn't involved in the development of Improvest, but I think it was another company with my collaborator and friend Michel Bonneau at INRA. Where back in 1994 we were testing out a vaccine as well, and looking at levels of boar taint related compounds in animals that were treated with that vaccine that they had. Now onto dietary strategies. So dietary effects on boar taint, because skatol, as I mentioned, is produced from tryptophan. You can manipulate that by using fermentable carbohydrates in the diet. That's the best approach. What it's thought to do is to decrease the available tryptophan by decreasing apoptosis of intestinal epithelial cells. So you're decreasing cell turnover, so you're then decreasing the availability of tryptophan, and you can actually lower skatol levels quite dramatically. So skatol really isn't a problem if you want to try to control it, but you can do it by diet. Androstenone, on the other hand, is really affected by sexual maturity. So if you want to control androstenone, you really have to have an animal that is immature. So you can slaughter at later weights, but that really, economically, that's a problem, right? So really, I guess overall, diet's not an effective solution for taint overall. It is for skatol, but not for androstenone. So here's another, so we actually got an idea for doing this. Actually, Phil Duke from Illinois, who's a reproductive physiologist, contacted me at one point, many years ago, sent me a snail mail letter, and that was all handwritten, and suggested that I look at intrahepatic circulation. And he included some papers that he had done, where what he'd done is actually induce the metabolism of boar taint of steroids in the liver. So let me just walk you through what intrahepatic circulation is. It's important in recirculation of steroids. So essentially, steroids are made in the testis, they're metabolized in the liver, and excreted in the bile as conjugates. So from the bile, then they go into the intestine, where they can be deconjugated by microbes and then taken up again. So you get this circulation going around, and it's either metabolized again or stored in fat, right? And that happens for all steroids. Estrogen, for example, is a classic example. So the idea was, can we manipulate the system here by altering gut bacteria or using binding agents? We did some work a while ago playing around with different antibiotics and whatever, but binding agents, what we thought was an interesting thing. So the whole idea is to block this deconjugation so that then the steroids are then excreted and then not recirculated. So I thought, well, that's a great idea. We played around with it for a while, and then I had this student, Kimberly Jen, who I got on this project. And to be honest, I thought this was never going to work, but it did. And it just goes to show you that you know, if it makes sense from the physiology point of view, it's worth a shot. So what she did was using various dietary binding agents, and the prototypical one to use was activated charcoal. Right? So if you feed activated charcoal to a dog who's eating something nasty, for example, you know, then you can absorb that. So she started off using a variety of different binding agents here in vitro. So in the lab, she checked the effectiveness of different binding agents to bind boar taint compounds. And then from that, selected activated charcoal and also tween to actually run feeding trials. And here's what the feeding trial looks like. So she was feeding a diet supplemented with either activated charcoal or tween for 28 days. And then you take them off that, and throughout this, you're taking fat and blood samples to measure androstenone. So this line up here is the control. So by the way, the cutoff for androstenone for acceptability is around 1. It varies depending on who you're talking to, between 1 and 0.5. So you see they all start off at an unacceptable level. The controls stay high, but when you feed activated charcoal, the level goes down over time. and it stays down for a while after you take it off, take the animal off activated charcoal, and then goes back up again. So it really shows that by absorbing the metabolites of androstenone in the gut, that you can actually decrease the amount of boar taint by actually increasing the excretion of androstenone. So we thought this was pretty cool. Where we are with this right now, a student of mine, Peter Park, is actually looking at more cost-effective and more realistic types of dietary additives, and with I think was actually quite good results, and you guys will be hearing about that in the near future. So I think dietary strategies for controlling boar taint are actually quite possible. Lastly, I want to talk about genomic selection, because I think genomic selection is really the final answer, right, for the problem. And when I talk about genomic selection, I think about the idea that we've learned all this work about all this information about metabolism. How can we use that to move into genomics? And because I've been working on this so long, I've actually seen the field change from metabolism studies to genomic studies. Because once you know which enzyme is involved, you can then look at the sequence of the gene that codes for that and look for variations that you can use for markers. So we did some work looking at differentially expressed genes in high and low levels of boar taint. We then looked for polymorphisms in candidate genes, initially by cloning and sequencing those particular genes, cloning by PCR and then sequencing. Later on, when the NCBI databases were built up, we could actually mine the database in silico. And for the next work that we're doing, we're actually going to be getting into transcriptomics and next generation sequencing to get more data. So here's some of the first papers that we did looking at differentially expressed genes. This was back in 2004, a master's student, Jennifer Stewart. We're actually using human microarrays here to look at differentially expressed genes. So this was back in the day before there were pig microarrays. We did this here, and we had a list of differentially expressed genes, and then later on we actually used a pig microarray to look at pigs with high versus low levels of boar taint. So from that, and from the literature, we were able to come up with a list of genes to look at. Oh yeah, and then some very early work, even before we had that, we knew from our studies that there were some key enzymes, like for example, cytochrome B5, SALT1A1, CYP2E1 that I mentioned before, and also 2A6, or actually 2A19. And a postdoc that I had, Zihong Lin, actually did a lot of sequencing work here to find polymorphisms in all 4 of those particular ones, and we still actually use these markers now. So this was very early work back in the early 2000s. Again, back in the day before there were really good databases available of porcine's genes. So where are we right now? Right now we have a SNP panel of about 100 markers in about 40 candidate genes for boar taint. And we're validating these in about 3,500 purebred boars. That work has already been done. I want to mention that this work was done actually in collaboration with Canadian Centre for Swine Improvement, the Ontario Swine Improvement, the CDPQ in Quebec, and with the Western Swine Testing Association in Alberta. So having all of the industry people on board made this possible. There's no way I'd be able to get out and get samples of 3,500 boars without having industry support. It's just impossible. So it just shows you when we bring a project to a certain point, having industry come on board and really help us out and get samples and work with us, it really makes all the difference in the world. So I'll show you some data. We got some actually quite nice results from this. So the macro effects were evaluated on androstenone and skatol levels of fat. And because we have EBVs for economically important traits, we can actually then check and see if any of the markers have any negative effects on these traits as well. So this work has been done, and I'll show you some data. And right now, we currently have a trial going on where we're actually validating the markers in a 3-way commercial cross. So the final cross that you normally have. So that's Duroc sire and Landrace cross Yorkshire dam. So this shows you the idea of what some data looks like. This is a relationship between the number of unfavorable alleles and the levels of androstenone in Duroc boars. So this is androstenone levels in fat here. And this is a bar graph that shows the levels of androstenone that you have based on the number of unfavorable alleles in those genes that I was talking about. So if you have more than 15 unfavorable alleles, you have fairly high levels of androstenone. The percentage of animals that's above an acceptable limit is about 75%. So 75% of the animals are tainted. If you only remove 5 SNPs you're down to an average value that's below the acceptable level, and you have overall about 15% of the animals are tainted. So this is a very powerful tool for selecting against boar taint in Durocs. And so we're quite excited about this, obviously, and it actually shows that selection against boar taint is quite possible. And I said my goal is to produce boar taint free lines of pigs before I'm done. So I'm not quitting today. Okay, I want to just sum up quickly now, just to kind of talk about it kind of in hindsight, how technology really drives a research program here. I mentioned about the test for boar taint, because you need to be able to measure boar taint if you're going to study it. So you have to be able to measure it. So the first test that we had that worked for a while. Then we had an ELISA, and then now we're moving on to aptamers. So that's going to just make things better. With our ELISA, we do our ELISA in 96 well plates. So we do 96 samples at one go. While before, doing one sample took a few hours. I moved on, so the whole idea of using primary cell cultures, I mentioned that already, using hepatocytes and Leydig cells to actually study boar taint metabolism in a more in vivo situation. I mentioned already about cloning and expression of nuclear receptors. I never showed you about reporter assays to determine ligand specificity, but that's the work I picked up in 2003 from my sabbatical. Again, the availability of sequence databases for porcine genes. The 100 SNP panel that we have now, I actually have expanded to 1,500 just by mining the databases. We're waiting now to validate those markers. High throughput and low cost genotyping methods. When we first started genotyping, it was one SNP at a time, whatever. Now we use a sequenome system. We get a SNP for about 25 cents. That's how much it's gone down. We'll be developing low cost SNP chips as part of what we're doing. The other really interesting thing, and this is kind of not really high tech, but it's a biopsy tool that we have. So we saw a biopsy tool that was published online. Actually, it was a paper by Christine Bays, who we just hired a while ago. They used a rabbit stunning tool, modified it to put fat biopsies. So we convinced them to send us one, actually as part of a collaboration. When I got it here, I brought it over to our machine shop, and they go, oh, no, we can do better than that. And they built a much more improved version, made 4 copies for a fraction of what it cost me to get the one from Switzerland. So it again shows you how if the technology is not there, you can take it and you can actually improve it. So this was actually very cool. And this biopsy tool has been really instrumental in the latest work that we're doing, where we're doing large scale validation studies. So we can pull samples on live boars without a problem. Right? When I say we, I mean people like Chris Crump and those kinds of folks. Right? Not me. Anyway, just again, I just want to give you a list of sort of grad students and postdocs that have been in my lab, and we have a few people here that are still working on this, and these are just the grad students that have been working on boar taint. I have other students as well who've been working in the poultry area. And then a little list on U of G faculty. I know I missed a few here. National international collaborators, I probably missed a few here as well. But again, it turns out, so Michel Bonneau and Kirsten Lundstrom are essentially the godfather and godmother of boar taint. And I met them in 1991 at a meeting in Ottawa, and they've been good friends ever since. Ilya Zamoretsky published a lot of papers. She actually was trained in my lab as well. Anyway, very interesting when you collaborate with people with a lot of different expertise. And I want to give special mention to this one person, Yamping Liu. She's the lab mom, technician, jack of all trades, equipment repair expert, student mentor, and this is Yamping. So Yamping. Yamping walked into my lab, 93 was it Yamping? And said, I'd like to have a job. And I said, well, I'm sorry, I don't have any money to hire you. So she said, well, I'll volunteer for a while. 2 weeks later, I hired her and paid her her back pay. And she's been with me ever since. So thank you very much. Yamping just holds it all together. I couldn't do what I'm doing, any of this without her. Just again, sum up funding boar taint research, basic and government work, insect discovery grants, so MAP for animal program. I had funding from Ontario Genomics, Ag Canada, and cluster work, whatever. Industry and applied, PIC and Genus did a lot of work with them on developing markers. I did a lot of other work with various drug companies who are all interested in controlling boar taint. And I mentioned already about our really important work with CCSI, OSI, CDPQ, and WISTA to really run our validation trials. So I can't emphasize enough how important it is to finally work with them. And Brian Sullivan in particular was a leader in actually going out and really getting the funding and really kind of taking me under his wing, or taking the technology under his wing to say, this is an important thing and we got to make it happen. So I'm really thankful for that. Okay, this last slide just to say where we're going now rather than kind of just end it. We are investigating some novel aspects of boar taint metabolism. Christine Bone, who's here, is going to be a new student in September. She's interested in looking at factors that affect the accumulation of androstenone in fat and transport into the fat cells. Evaluating more cost-effective feed additives for controlling boar taint. I mentioned that's Peter Parker's work. We're actually expanding the genetic marker set for taint. I said now at 1,500. And validating this, and actually Holly Archer, who's here, is she'll be presenting this afternoon. We'll present some data on the marker set we have to date. And the other thing that I got involved with is once we get into genomics, it really doesn't matter what the breed is or what the animal is. It's really all the same technology. So working with Flavio Schenkel and some other folks, we got into this idea about looking at genetic markers for ketosis in dairy cattle, and so we're pretty excited about getting into that one as well. So with that, thank you very much.
Perfect. Thank you, Jim, for that fantastic presentation and very impressive research program. Are there any questions?
Yeah.
Excellent. How important of the microbiome,
we haven't done anything to do with the gut microbiota. Clearly, it's going to be important because the gut microbiota makes skatol, right? And so you can manipulate the gut microbiota and then decrease skatol levels. But we really haven't done the kind of metagenomic work, for example, to really nail that down. No, one of the things that we've done with boar taint is really stay with the pig model. Yeah.
If you reduce skatol and androstenone, is there any impact on fertility in the horse?
No, because if you remember when I showed you the pregnenolone either going to androgens or to androstenone. So if you manipulate the pathway so you want to actually just shut off one branch and not the other, then you don't affect, as long as they're making normal sex steroids, you're good to go. Some of the initial selection against androstenone inadvertently reduce the levels of sex steroids as well. And so clearly you're going to have a problem there. So that's why, and I didn't say this, but that's why genetic markers are really useful. Because if you get specific genetic markers that only target that one part of the pathway, then you can really knock it out specifically. Yeah.
Were you ever asked what, if we did get it, that it was a simple way to wipe out the protein, what would see in the industry and not having to
well, Case DeLange and I've published a bunch of stuff on that. I think it's similar to using, well, it would get similar results to what you would get from using a lot of other technologies, beta agonists, for example, or growth hormone. But you do it without any of the negative impacts on it. I think if I remember the number, it depends on the margins, where you are, but it's like a 40% increase in productivity. It's really quite significant. Yeah. Sorry, me?
Yeah. I really mentioned really showing off, background research, implication for vascular new pathway. But secondly, two questions. Number one, and it's more important for you all generally develop the amount of lines that are actually loading more than start with the sex hormone. More important for application in the industry is aggressiveness. Right. So I think aggressiveness will be important in accept that.
Yeah.
Just let the line go important, smell, but also milking has to be less aggressive.
Yeah, there's no question.
That's very important animal welfare consideration.
Yeah.
That has to be considered number one.
Yeah.
Secondly, what's your opinion in terms of newly emerging CRISPR technology, gene editing?
Yeah.
Could actually eliminate that gene actually without even a GMO issue.
well,
Last time, so
okay, so let me start off by talking about the aggressiveness, and there's no question that boars are aggressive. Well, pigs are aggressive anyway. If you mix any pigs, they're going to fight. I think the idea is that once they establish their own hierarchy, they settle down a little bit. But clearly, as you select for low boar taint pigs, you need to also make sure that you keep an eye on aggressiveness, so that's clear. As far as CRISPR technology or any of those genome editing tools, whether or not they're accepted as non-GMO is another issue, right? But there's no question you can edit them. In fact, we're doing some work to actually check that out. But we don't know where it's going to go as far as being acceptable or not. But yeah, the whole idea with genome editing is that you can introduce a mutation wherever you want, right? So based on what we know, we could argue that any mutation that we insert could be there naturally. We just haven't found it. That would be the argument. Now, whether everybody buys that or not, I'm not sure. So that's a whole different kind of story. So, yeah.
See, the patent crops,
Yeah.
recent news in the US, actually FDA, reviewed the new variety crops by CRISPR technology,
Yeah.
and now regarding the GMO.
Yeah, well, the plant world has been much more accepting of transgenic technology than the animal world, for sure. Yeah, I think there was a question up here. Sorry, using what? I never heard, can't hear what. I'm sorry, I don't understand the word. Oh, zearalenone, sorry, yeah, okay. Sorry, it's an estrogen. No, I don't think estrogen is really going to help. Boars make a lot of estrogens already. One of the feed additives we're looking at is called a spent filter rate. So it's a diatomaceous earth that's used for filtering corn syrup. And that's the one that we're really excited about because not only does it bind protein steroids, but it gives you energy and protein as well. So it's a low cost, you can actually replace corn in the diet. So we're playing around with that, and we're pretty excited about that one. Oh, yeah?
You mentioned that human castration is not well accepted in America and Europe. What should be done to that technology?
Well, so the question was, what should be done with immunocastration technology to make it more acceptable? Some of it's public perception, as far as I know. I know EU is totally against it, right? I think in some countries, like in Brazil, for example, they're vertically integrated, they use it a lot. I think it's starting to become more acceptable in Canada and in the US. There's a cost, of course, right? So that's part of it. And then you also have to get 2 shots of the vaccine to the animal. So there's a lot more handling involved. But I think on the short term, immunocastration, I think it's the greatest thing ever because it'll get the industry used to using boars. So that when we then can breed the boar taint out, then we just stop using the vaccine and we're good to go. Because I think the biggest problem is transitioning the industry, right, to anything that's new like that. Because it has big implications in organizing the slaughter plants and yeah. Okay. Sorry.
What does it have to do with the slaughter plant who now says it now has acceptable, we don't pay very much for that. So is there something in place here in Canada where we say, well, these animals are allowed to show up at the slaughter plant if that's really because?
As far as I know, and maybe Rod is here. Rod can tell us what happens to the boars. But as far as I know, a lot of them you were usually shipped to the U.S. And I mean, probably a lot of them are late castrated too. Not supposed to be, but. Okay, thank you.
All right, so next on the program is the poster